Hello, everybody, and welcome to the first episode in my course entitled General Science. In this course, I want to talk about all the different branches of science and some of the basics of each one, just as kind of a general overstudy of science in general. In this very first episode, we're going to talk about what science is and the scientific method. The scientific method is how scientists actually do science. So if you wonder what a scientist does in his day-to-day job, he or she does the scientific method. Okay. Now the scientific method has several steps that you kind of have to follow, so that you can investigate something you want to learn about. There are a few steps, and we're going to look at each one and kind of talk about each one individually. The steps are: you have to have a problem or a question you want to answer. So, for instance, maybe a question that a lot of people might want to answer is, how do we solve climate change, or how do we stop climate change from occurring negatively to our country, or to our world? So that could be a problem or a question that a scientist might look at. The next step after that is to do some observation and research. You need to go out into the field and you need to take samples, and you need to observe the weather and observe all the different goings on the planet. You need to do some research. You need to read some books. You need to probably go and do a little bit of web searching. You need to find out what's going on already. What do we already know about this issue? Because if you're trying to study something and it's already been kind of figured out, then you're wasting your time. So you want to make sure that you know what's already known so that you can learn something new. Then you want to formulate a hypothesis. So now that you know all these things about whatever it is you're researching, you're going to want to be able to kind of come up with a statement basically saying what you think needs to be done. So for climate change, a hypothesis could be, if we lower carbon emissions by 50% over the next 2 years, then that will benefit the global climate. That's a hypothesis. Usually they're an if-then statement. So if I do this, then this happens. Then you're going to want to perform an experiment. So you're going to want to design some kind of way to test your hypothesis. So in that case, you could create like a simulation, or you could create like a special room or like box with like an environment in it, and you could try to take CO2 out of it and see if that affects the climate in that box, something like that. Then after your experiment, you're going to want to collect and analyze the results, see what happened, right? So you want to make sure there's some sort of measurable result that you can get out of your experiment. In this case, it would be like, well, did the climate change when I took the and you might want to have certain parameters you're checking, like what happened to the amount of water in the air, what happened to the amount of oxygen in the air, like that kind of thing, something you can actually study. And then from those results, you can draw a conclusion. You can say, well, I took the carbon dioxide out of the system and all the plants died. Oops, I didn't do my experiment very well, so I have to try again. And so after you're done, the most important step then is communicating the results. So once you do your study, you have to be able to communicate to others what you did. Oftentimes, what scientists do is kind of shrouded in mystery, and we only really ever see the end product. You only ever see it turns out that you can drink a glass of red wine every day and you'll be okay. Or it turns out you shouldn't swallow toothpaste or whatever, right? You see like those BuzzFeed clickbait articles about, scientists figured this out. Well, that's the end, and that's not necessarily good communication of results either. Those are usually pretty misleading. So we're going to talk a little bit about how to do that better as well. So let's move on. We're going to talk about each step in an example. So we're going to talk about baking bread. So here's the example. We've got a guy named John, and he's watching his grandma bake. And he asked his grandmother, okay, why does the bread rise when you're baking it? And she explains that yeast releases a gas as it feeds on the sugar. So that was grandma's explanation of what was happening with her bread. Okay. So now John wonders, okay, if it's feeding on sugar, if I change the amount of sugar, is that going to affect the size of a bread loaf? Obviously, yes, but let's let John figure it out himself. So he's going to design an experiment based on his question, right? His question was, if I change the sugar in the bread recipe, will the bread rise differently? That's a good scientific question he didn't know the answer to, so he's going to figure it out. So now for observation and research, John's going to go online, maybe check out a book, and he's going to research baking and fermentation, and he's going to try to come up with a way that he can test this question accurately. So he's going to come up with an experiment based on his research, right? He observed his grandma baking bread already. Now he's going to do a little bit of reading. He's going to learn a little bit more about the process of all of this, and then he's going to come up with an experiment to test this question. So first, though, he's going to come up with a hypothesis. And he decides that his hypothesis is going to be, if more sugar is added, the bread will rise higher. That's a pretty good hypothesis. It's an if-then statement. If he adds more sugar, then the bread will rise more. That's a solid hypothesis. And it's something that's testable, too. So he can actually go and make bread and add more sugar and see if the bread gets larger. So that was his hypothesis. And now we're going to actually set up his experiment. So for his experiment, he needs a control group. Now, in an experiment, generally you have what's called a control group. Control groups are your normal conditions. So a control group in John's experiment would be that he needs to bake bread using the regular recipe his grandma uses. That could be his control group. And then anything that he does differently would be then the experiment, right? He needs something to compare it to. If he doesn't have anything to compare it to, his experiment would be meaningless. It'd be like, well, I put this much sugar in this time and this much sugar in this time, and this one was more. It's like, yeah, but is it more than what's normal? So you need to have something that's normal to compare it to. That's a control group. Some scientists call it the standard of comparison. So what are you comparing your new experiment's results to? And he's setting up a standard, and that standard is his grandma's recipe. So once he has his control group set up, he's going to actually do his experiment. Now, when he does his experiment, he needs to make sure that he's only changing one thing at a time. So when he's baking his bread, he needs to make sure that he is only changing one thing. So in his experiment, right, because he's dealing with sugar, he's only going to be changing the sugar in his loaf of bread. He's not going to change the flour or the eggs or anything else he puts into his bread. He's only going to be changing the amount of sugar. If he doesn't do that, then it could be some other factor that's making his bread different than the sugar. So he's got to make sure that he's very accurate with his other ingredients and that he's using the same amount each time. Also, when you're performing an experiment, having more data is always good. Now, when you're younger, when you're in middle and high school, generally we settle for at least 3 trials. That means you're going to do each experiment 3 times. And the reason for that is just so that you don't have an oopsie, I guess, or a goof up, and your one trial at whatever amount ends up behaving quite differently than it should. So you're basically trying to make it so that you don't get some sort of outlier data, some data that doesn't make sense being added in. So if you have 3 trials, that one trial that was really different, you can be like, oh, that one probably we screwed up on. That's probably not correct. We can go off these other 2 that seem more in line with what's going on. So if we take a look then, I've got Johnny's data table right here on my computer. So he changed the amount of sugar, and he does several different amounts of sugar. Okay, and just to summarize it for you, right, when he puts in 25 grams of sugar, he gets roughly 758 cubic centimeters of bread. When he puts in the 50, his control group, right, the amount his grandma uses, his bread is about 1260 cubic centimeters. And then as it goes up, the size of the bread actually decreases. So he's finding out that maybe his guess was wrong. If you put too much sugar in, it's not going to make your bread any larger. So that might be confusing for him. Based on what he knew, he would expect the bread to get larger. So Now what's he going to do? He's going to have to analyze that data, those results that he got. So he's taking a look at it, and he notices that his control was the best, but it wasn't a lot better than the 100. If you go back to the data table, okay, it was only about 60-ish, maybe, okay, like almost 100 cubic centimeters bigger. That's not that much different. So what he's going to do now is he's going to take a look and say, well, his original hypothesis didn't work, right? You put more sugar in, it doesn't necessarily mean you're going to get bigger bread. So unfortunately, you can't just throw sugar into bread mixture and then you'll get bigger bread, right? We could solve world hunger in a couple days if we could just make ginormous amounts of sugar bread. Okay, but now he's going to retest and see which one's the best in between 50 and 100. So there's some wiggle room there, and he wants to see which one works out the best in that range, because clearly there's this area here now where you can make bread larger. So he's going to do some retesting. That's the conclusion, is that he can do better. He's going to try his experiment again. So now he's going to gather his materials, and he's going to do it again. And this time, he finds out that, once again, the 50 and the 90 are a little bit off, but right in the center of that range, at the 70 grams of sugar, his bread is way bigger, about 300 cubic centimeters bigger than it was normally. Now, because of that data, he can say conclusively that not only does he need to add a little bit more sugar to his grandma's bread recipe to make it larger, if he wants to, right, that's going to be his ideal amount of sugar in that range that he needs to use to get a nice big loaf. So now his conclusion then, at the end of the scientific method, at the end of his experiment, is that 70 grams of sugar is what produces the largest loaf. So he managed to get his original hypothesis correct. He did have to increase the sugar. It's just not as much as he had originally thought. He originally increased it by 50 grams and realized that was too much. And so then now, when he increased it by 20, it actually worked. So he did end up actually proving his hypothesis correct. He added 20 grams of sugar, the bread got larger. So now, to communicate his results, John, in this instance, right, the person he needs to communicate with is probably his grandma, right? Hey, grandma, your bread could be bigger if you use 20 grams more sugar. And then she might be like, oh, thanks, John, because grandmas do that kind of thing. And then she'd make bigger bread for the rest of her days. John could also talk about his science class and be like, hey, like, look what I figured out, and share his results. Now, how scientists do that is a lot different. What scientists do is when they finally finish something, they write it up in what's called a scientific paper, and then they have to submit it to peers to review. So there's a very extensive peer review process, and what that means is that other scientists that are experts in that field who have studied it before have to look at their lab, their experiment, their results, and then they get to say, I don't think this is legit or not. And so several different people get to take a look at it and kind of call scientists out on their work before they get to publish it. And then if it gets through that process and it's still okay, then it gets to be published in a journal and other scientists can read it and learn, and goes on. And at that point, right, some newspaper picks it up or some magazine, and if they, summarize it poorly and put it up in big letters, and everybody goes on BuzzFeed and goes, oh my gosh, you mean I can eat 50 cookies and be okay? I won't gain weight? And then they're wrong because they didn't say exactly what it actually, you know what I mean. So the scientific method is how science is done. And it's important, I think, not just for a science student, but for everyone to understand this process. The reason being is, especially now with the pandemic going on, for the first time for a lot of people, they're seeing the scientific method play out in real life, right? The disease started. We're trying to make a vaccine. We're trying to treat it. We're trying to figure out what to do to minimize the risk. And a lot of science in that beginning phase is guesswork. It's, okay, do we do this? Let's see if it works. Okay, it didn't work so well. Let's try this now. Okay, that worked better. What about we do that and this? Does that make that better? No, it didn't. Okay, we need to try this again. Same thing with a vaccine. Okay, we tried this one. Oh, that one's not so great. It has a bad side effect. Let's try this one instead. No, that one doesn't cure the virus. It takes a while, and it's frustrating, I think, for the public who doesn't understand that there's a lot of wiggle room in this process. They're used to just seeing the results, and they're not really used to seeing the scientists struggle for years to get to that final result that they get to share with you. And so, as a regular person, if you're not a scientist, if you don't have that knowledge of a scientific method, you're just going to get frustrated and angry that they haven't come up with a solution already. And that's just not how it works. And so it's important to take in mind that the people who are trying to cure this virus are doing their best, and they are working as fast as they can within the parameters of how science is done. And one day they will figure it out. But until then, you're going to have to bear with them. Just like any job, if you want to do it right, it takes time. Thanks for watching my first episode in general science. Like I said, this is about the scientific method, and we've got all sorts of awesome steps for that method. Remember, here are the steps in order one more time. You got to have a problem or question. You need to observe or research about that question. Formulate a hypothesis, so make a guess as to what you think might change your results. Do an experiment to figure out if you're right. Collect and analyze the results of that experiment. Draw a conclusion. Were you right? Were you wrong? How can you improve? And then communicate those results. Thanks so much for watching. See you in the next episode.